today's class we will discuss about DNA replication. DNA is a molecule which can produce its own copies. Scientifically speaking, we give the terminology replication. In fact, DNA is getting synthesized. Let us see that at what time the DNA get replicated in the cell. Even if it is in the microorganism like bacteria also, the same DNA replication takes place. We have studied in class 11 the cell reproduction. At that time we have studied cell cycle. In cell cycle we have studied three phases, G1, S and G2. When we have seen G1 as gap 1 or growth 1, we could see that the synthesis of proteins takes place. But when we have seen the S phase, that is the synthesis of DNA. So during S phase of the cell cycle, the DNA synthesis takes place. So always remember the DNA replication or DNA synthesis takes place during the S phase of cell cycle in a cell or in the case of microorganism. So we will see the introduction of the DNA replication as well as the experimental proof of DNA replication as a semi-conservative mode of DNA replication. Let us go for the first part, the introduction of DNA replication. This is what we see in a parent DNA. Suppose this is a cell. This is a parent cell. This parent cell has got the DNA strand inside the nucleus. This is the DNA strand which you are seeing in the parent cell. So this DNA has got two nucleotide strands. And the first nucleotide strand is with adenine, cytosine, thymine and guanosine in one strand. And the complementary other strand is having thymine, guanosine, adenine and cytosine. And in the first step of the DNA replication, the hydrogen bonds between these complementary nitrogen bases will break. When the hydrogen bonds break, normally you see these two strands will be separated. So these two separated strands are shown in this blue color. And now you don't say as parental strand, you can call it as template strand. So this template strand will guide or will decide what should be its complementary strand inside the nucleoplasm. All these processes are taking place in the nucleoplasm. So nucleoplasm is a pool which contains so many nucleotides. So from the nucleoplasm they get their complementary or corresponding nucleotides. For example, suppose this blue color strand is acting like the template strand and adenine is the nitrogen base. So from the nucleoplasm they will get paired with the thymine. So a hydrogen bond formation takes place. In the same way, cytosine will get complementarily paired with guanosine. Like that, all the nitrogen bases will be paired with their complementary nitrogen bases which are selected from the nucleoplasm. The same thing repeats in the other separated parent DNA strand also. As a result, from a single parent DNA, you are getting two daughter DNAs. Now, when you analyze these daughter DNAs, you can see that in a single daughter DNA, 50% is coming as a new polynucleotide strand which is as represented in this arrow, and the next 50% is coming as the parental strand. What does it indicate? Out of the two strands, one is conserved, one is preserved. So you call it as conservative. Since 50% is conserved, you can give this as semi-conservative. So what's an trick? In 1953, they proposed the double helical structure of DNA. They had also given a light or an additional opinion that there is a chance of giving multiples. There is a chance of giving replicas. And they also added that these replicas will preserve 50% of their parental strand, and the new 50 will be the new strand. So you can confirm that this is nothing but a semi-conservative mode of replication. So we come to the conclusion that the type of replication of DNA is nothing but a semi-conservative mode of replication.